We will continue our discussion about this VCAM-1 gene in the pancreatic cancer cells. So far in our previous videos we have discussed about the in vivo results of the effect of VCAM on the pancreatic cancer cells. That is how the pancreatic cancer cells react when the VCAM levels are up and down using the silencing RNA against the VCAM or overexpression using the plasmid against the VCAM. So in this video, our focus will be on the in vivo studies or the results that are experiment that is done on the mice model or the xenograft mice model. So in this experiment, what this researchers has done is that they have injected this subcutaneous injection of this pancreatic cancer cells, one with the over expression, one with the silencing of this VCAM-1 gene, and another one with the control silencing RNA. That is, here the VCAM genes are not inhibited. And in the case of SH VCAM-1 or silencing RNA against the VCAM-1, here the VCAM genes are inhibited. In the stable inhibitions of these VCAM genes has been obtained in the PDAC cell lines. So the black arrow represents the subcutaneous injection of this SHNC, or where the VCAM levels are expressed normally in the pancreatic cancer cell lines, whereas the white arrow indicates the subcutaneous injection of the pancreatic cancer cells with the silencing VCAM-1 RNA, or where the VCAMs are been silenced or inhibited. And what they have find out there is that after 28 days, the tumors with the wild type VCAM expressions has, or the subcutaneous injection point where the pancreatic cancer cells has the wild type VCAM expression levels has the high tumor size compared to the case of the pancreatic cancer cells where VCAM-1 is been inhibited. That can be seen in the panel A, and in the panel B we can see that tumor volume, where it is clear that the tumor volume size is lower in the case of this VCAM-1 gene is been silenced in the pancreatic cancer cells compared to the case where VCAM-1 gene is been normally expressed in the pancreatic cancer cells. And these things they have also validated using the RNA expression levels of this tumor VCAM RNA expression levels of these tumors, that is shown in the panel C, where we can see that the small tumor size or the tumors that are obtained from a position where this silencing VCAM-1 pancreatic cancer cells is used shows the low VCAM-1 expressions compared to the case where the normal pancreatic cancer cells is being used, or the control pancreatic cancer cells is being used. And these things are also been validated using the Western blot, where we can see that VCAM expression levels are low, where VCAM-1 is been inhibited in the pancreatic cancer cell lines. After that, they check the proliferations of these tumors using the markers, that is Ki-67 markers, and they can see that the proliferation is more in the control case where they have used normal pancreatic cancer cell lines compared to the case where they have inhibited the VCAM-1 genes. And these things are been quantified on the right hand side, which shows that higher Ki-67 expression levels in the control compared to the case where VCAM has been inhibited. And when they check the tumor volumes, they find the similar results in the tumor volume that in the control case where VCAM expression levels are not been interrupted or disrupted, they see that high tumor volume, whereas when the VCAM-1 expression levels are inhibited, they see the lower tumor volumes. And in terms of the weight also, they see the similar results, that is in the control case they see that high weight compared to the case of this VCAM-1 inhibited pancreatic cancer cells injection point tumors, where they see that the weight is significantly lower. So all this data indicate that VCAM-1 helps in the proliferations and growth of the pancreatic cancer cell lines at a much higher rate compared to, and VCAM-1 inhibitions can prevent this proliferation rate of the pancreatic cancer cell, of the pancreatic cancer tumors in the mouse model, in the xenograft mice model. So I hope you understood the final outcome of this experimental results. If you want to understand more about their experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.